Very good morning, students. Welcome back to lecture 11, graft versus host reaction, graft versus host disease, part of transplantation immunology or transplantation biology. The class is divided into several sections. In terms of, we will recap for the last week two lectures briefly in one or two slides. What is about fundamental concept of transplantation? What is a host? What is a recipient? What is the compatibility in terms of HLAs or MHC molecules? HLA typing, how different HLA molecules have been produced, polymorphism in per se. And then what are transplantation barriers in terms of phenomena of whether the host is going to accept the tissue donated by the donor or not. That is kind of a lot of transplantation barriers we have discussed. Two concepts we will highlight today's class. One is transplant rejection. One is graft versus host reaction, or disease, GVHD usually called. And general mechanism of donor and recipient crosstalk goes through adverse reactions. That may be a process of hyperacute reaction, acute reaction, or a chronic reaction, or chronic response. We will see that what are those in the class. Coming on to fundamentally, we should appreciate our own immune system to protect us from different pathogens and also make us healthy. At the same time, we have covered a lot of topics in terms of the B cells, basics of innate and acquired immunity, and also we have seen how antibodies can elicit complement system, antibody antigen reaction. We have seen inflammatory response. We have seen opsonization. We have seen in terms of the polymorphism how different antibodies can be produced by VDJ recombination. And in terms of HLA types, we have seen in the last class. So there are different genetic loci. There are different polymorphisms exist called haplotypes, given a variability between different population. So basically, coming on to that concept of immunology, the course, we have probably covered 30% or 40% of the concepts, and we have much to know, much more to explore. For example, we haven't been discussing or we haven't been talking about interleukins or interferons. What are those? And also in terms of the allergic reactions, in terms of the immunocompromisation, autoimmune diseases, how the system will help to kill its own cells. We will see into that in further classes. So coming on to organ transplantation. Excuse me. Organ transplantation is the transfer of a healthy organ or part of it from a donor to recipient, at the same time establishing vascularity. A lot of pioneering studies has been done in the organ transplantation, successful surgical techniques, and also advanced immunosuppression methodologies helped us to advance organ transplantation, make it a successful event, not to cause any cross reactivity. But at the same time, the worry is that there is a lack of organ donors. There is a proportion of recipients is very higher, exceeding the amount of the organ donors that exist. That is a part of concern for everyone. And we have been discussing about what are the tissues or organs that can be used for organ transplantation. And also, if you zoom in, each organ will have their own limitations or their own advantages depending on the recipient. Who is a patient who is suffering from a kidney disease, he can get a kidney transplant. That is the most commonly used. And bone marrow transplant are used in terms of the hematopoiesis defective, or in terms of the whole immune system of a recipient is very badly deficient or or compromised, or it's not doing a proper immune response. Or in case of leukemia, anemia, you go with bone transplant. And most difficult based on surgical and immunocompromisation procedure, heart transplants are the most difficult, and liver can be used for viral and chemical agents. Pancreas is as concerned, insulin production, diabetes mellitus, and common are skin grafts that used for the self grafts called isografts. We will see used to burn victims. The most fundamental mechanism of transplantation is a distinction from self and non-self. How an immune system, that is a fundamental protective agency for a human or for an organism, how it can distinguish self from non-self or foreign molecule. And if a foreign molecule, how it detects, how it destroys the foreign molecule. That is fundamentally important for transplantation studies, because most of the transplants are coming from different species or different organisms. So there may be a mismatch in terms of the immune system or immune cells. They may not be compatible, and we will see how is histocompatible and incompatible, how it causes or elicits a proper immune response, that we will see in this class. Coming on to recap in terms of the immune system, as discussed, anything foreign it detects and it destroys it, and anything self, non-foreign, it keeps it and it keeps at safe measures. And how it does do it? It is trained, well trained to distinguish self and non-self. A fundamental memory system exists in immune cells, and this is a self in terms of the recipient. The green is donor. Let's say they can each other recognize them as a foreign or non-foreign. And how the process happens? Basically, it happens through a specific molecules called histocompatibility genes, MHC molecule, major histocompatibility complex. They are always also known as human leukocyte antigens. They are capable of recognizing a self from non-self. And basically, we have seen that there are two classes of MHC molecule, MHC1 and MHC2. And MHC1 is basically present in every nucleated cells in the body. You can say a specific kind of immune cells that's present in the body. MHC class 1 are positive. But on MHC class 2, they are specifically expressed on antigen presenting cells called APCs. We have discussed it. What are those APCs? Monocytes, macrophages, dendritic cells, and B cells also serve as antigen presenting cells at a given scenario. So there is diverse cells, and the whole immune system can be distinguished and separated by two different molecules, MHC1, MHC2, for their self pattern or self recognition or self marks. So given the complexity of the foreign antigens, pathogens, given the complexity in terms of the immune system, how MHCs, only class 1, class 2, can recognize variable different recognition partners, we have been discussed that there is different loci, different genes, loci, that encode for MHC class 1 and class 2. There is different categories of class 1, class 2 gene families are there. Among them, we have discussed specifically on a concept of polymorphism, HLA polymorphism, that can have individual two species or individuals among the same species can be distinguished by HLA type they are carrying. That we have been covered in the last class. So basically, class 1, class 2, self, non-self. And if let's say class 1 recognizes a foreign, what does it do? Class 2 recognizes a foreign, what does it do? Basically, foreign means they have to destroy it. How they are going to destroy it? It's again common mechanism. We will see if it involves an antibody, it's nothing but whatever we have studied in the last class, antigen-antibody interaction, classical pathway, immune response, it recognizes it at a foreign antigen. Basically, a cascade of complement system, complement proteins will come up. So the final cascade, lytic pathway, a complex of complement proteins, C5, 6, 7, 8, 9 come and kills the cells to destroy the cells through antibody. Let's say how it happens through a kind of class 1 MHC molecule. So we will see that how it happens, but in fundamentally leaving the antibodies, class 1 and class 2, they are nothing but antigen presenting cells. They are mainly involved in cell mediated immunity, that is in terms of interaction to T site lymphocytes, basically T lymphocytes, right? We will look into that. We already looked in the pattern which T lymphocyte is class 1, class 2, and the ternary complex they will form. So we will look more into again further slides. This is the fundamental recap of MHCs, HLA types for transplantation studies. And we also looked in terms of organs available for transplant, a lot of donor and compatibility with the recipient, in terms of HLA typing, in terms of blood transfusion or blood matching system, we looked at all. And also two concepts we will study further in this unit 3. One is transplantation barriers, one is immunosuppression. This is we will see in the next class. What are the transplantation barriers? In most fundamentally, there is a process of rejection. What is the rejection? What is graft? We will look into slowly in further slides. So transplantation barriers are nothing but given the extent of natural polymorphism in a population, the donor and recipient will always differ for allelic variants of numerous proteins. That we already discussed, HLA haplotypes. Given the extent of natural polymorphism in a population and the polymorphism are different diverse HLA types available, the donor and recipient always will have differences, minute or major differences. If the differences are minute or minor, there may be another response from the host immune system. If the recipient and donor differences are major, the response will be a kind of drastic response because it thinks it's a kind of causing harm to the body. We will see that. So except given a situation of monozygotic twins, they will have almost all similar donor and recipient will disappear. They both becomes a donor, they both become recipients based on the almost genotyping, similar genotyping. So such differences in transplantation barriers, whatever that discussed, leaving the monozygotic twins, donor and recipient differences, that are differences are recognized by immune system. We know that the immune system is highly complex and highly organized. Whatever the small change in terms of foreign or non-self to self, they are able to quickly detect it with the shortest possible span of time, and they will elicit an immune response based on the recognition, based on the kind of whatever that coming to the body in terms of the graveness or issue to the one's body. So one of the biggest barrier in terms of the transplantation barriers is nothing but the immune system itself, right? Being the highly organized, highly complex and highly evolved, the immune system itself is the barrier because you cannot bribe it. Basically, it's one of the highly disciplined system, and each one of the system or each one of the cells have their own autonomy, their own self recognition or self gene regulation, their own molecular pathways. At the same time, a lot of crosstalk happens among the immune cells. And if you try to bribe the system, it will not accept. You cannot bribe 1,000 cells at a time. You can bribe one B cell, but there are 10 power 5 B cells in a given system. You can bribe one T cell, but there are tons of MHC molecules that interact and replace with the T cell. So it is a highly complex, highly organized, at the same time highly disciplined. So this system itself causes one of the biggest transplantation barrier. If it's not like that well organized, you could have simply replaced with many tissues from donor to recipient, right? Anyway, the immune system is, you can bribe it quickly, bring a donor with some kind of cash, some billions and billions of dollars, immune system will be compromised, it's okay to take the tissue. It will not, it will never accept that. Anything foreign, whatever the minute, my duty is to kill you. I will go with absolute sincerity to kill you. Don't worry about that. So that's a way immune system. That is one of the difficult barriers for transplantation studies. Different cells, different tissues, autonomy itself. APC, they have their own autonomy of gene control. In terms of T cells, they have their own autonomy, B cells and tissues, even the lymphoid organs. So there is highly autonomous organization at the same time. Highly crosstalk. If you break this system, it can crosstalk with alternative redundant pathway. It can again elicit the same response. So that is one of the biggest barrier. You cannot kill the whole immune system to compromise something. But if given a grave situation of the patient dying with the kidney disease, you have to do it. By the way of immunosuppression. Basically, you will kill it with some kind of chemicals that we will see. But in a given normal scenario, immune system will not compromise. Thus, the immune system, one of the biggest barriers for transplantation studies, is a highly evolved, elaborate, and effective mechanism to protect the organism from attack by foreign agents. And this same mechanism of protection causes rejection of grafts from anyone who is not genetically identical to the recipient. That's basically grafts. They are foreign, nothing but foreign. So, given a scenario, so they are going to, immune system itself recognizes and destroys. So coming on to fundamentally little bit, because a lot of new terms will come up in this chapter, you have to make notes again for this. So basically now you have to go back in fundamentals, talking about a terminology that is called grafts. Overall in the unit 3, overall 1.5 months, we are first time speaking of a term called grafts. So just recap our basics. What is grafts? What is a graft? So if you think of grafting process, you first bring up similar to fundamentals of plant systems, and there is skin grafts in animals comparatively, you see, but mostly studies were done in the beginning, pioneering studies, and not the human tissues. Plant tissues are well known for grafting. You're given a tissue 1, tissue 2, plant 1, plant 2, you just make a specific incisions, that just like a lock and key model, you put the incisions together, you tie them, you wax them, you leave it. At a certain time point, that both plants will combine together, the plant becomes normal growth, and this is a process of grafting. A shoot or twig inserted into a slit on the trunk or stem of a living plant from which it receives a sap is called a grafting process in plants. At the same time, another abbreviation definition for it is that a piece of living tissue. See that, a piece of living tissue that is transplanted surgically. Oh, it's also called as a graft. And the graft, in terms of transplantation biology, specifically alternative synonyms are, it is also called as transplant, implant, and the process of grafting or transplanting in humans or other organism is called transplantation. That's a fundamental definition. So basically, you are going two different things or one different thing, you are grafting it. So let's take some time go into visuals here, because most of the topics is always the concept is, for example, either graft or the host, either one of them fighting with each other. When one dominates over the other, they will use same immune mechanism, same immune cells, nothing but different. So graft will not have different immune system completely coming from space, coming from aliens, or the host will not have complete unique. Both will have the same immune system, same immune cell. So it will be very confusing unless you don't distinguish which one you are talking, which one is attacking. So I made a graphical representation of this so that you can have a kind of a mnemonic or imprint in the brain, so you can see that which one is killing what, which one is dominating, which one is immunocompromised, which one is causing the problem. So it is always fundamental to this chapter of transplantation. If you don't know that, you are talking about graft attacking the host or the host attacking the graft in terms of immune cell, you will be completely confusing. So look into this kind of color code. We have a color here that coming from the graft. This is the graft cells coming from the donor in terms of transplantation. In terms of the host, this is blue. Host is a recipient. So a graft comes from the donor part of the graft. Host will have their own host cells or system. Then it undergoes a process of transplantation or grafting. Then the resulting product is called a transplant. Resulting tissue is called transplant. Donor, graft, recipient, host, transplantation, the transplant will come. That's the basic fundamental, looking at two different tissue types or cell types. But at the same time, we need to talk about much of the immunology as such. Most of the concentration of the topics will be on immune system and how immune system is going to reject the graft or accept it. So basically now you need to bring two types of cells here, coming to graft and host here. You need to bring their own immune system. Graft will have, because we have been discussing, it's a living tissue, right? Grafting is a living tissue you are bringing. So it should be having some living cells. Most probably the immune cells will be there from the graft, and the host will have immune cells. So think of a scenario, a transplant coming from this. Think of a scenario of the immune cells. You might have a timer of these immune cells, right? We will see further. But coming on to just looking into the graft in their immune cells, leaving the immune cells, just look into the types of the graft can be distinguished into based on the tissue where it is coming from. We will see mainly they are differentiated based on the graft. They are called four types. That is nothing but autograft, isograft, allograft, and xenograft. This is very fundamental. You might get a lot of 2-mark questions on that. So based on the graft coming from where, leaving the host, the grafts can be distinguished into four types: autograft, isograft, allograft, and xenograft. Take them individually. Autograft is a self tissue you are giving from. Nothing but you cannot distinguish the colors. You are taking one tissue from my body, the skin. I'm putting the skin into another portion of the body. That's called nothing but I'm grafting my own tissue. That is called autograft. The self tissue transferred from one body site to another in the same individual. Transferring healthy skin to a burned area in burn patient is usually a kind of example of autograft. Isograft is a tissue transferred between genetically identical individuals. Basically, I'm saying the haplotypes of the two individuals is completely identical. There is no minor difference, 0.001, complete 100% identical. So those are called as isografts, nothing but in humans. They are called monozygotic twins coming from the same genetically identical. So they have same haplotypes. So then you can easily transplant kidney from a twin to the other, given a condition of monozygotic twins only. At the same time, allograft, this is the most studied, most important mechanism. An allograft is a tissue transferred with genetically different members of the same species. For example, among humans, for example, myself and other, I'm in need of some kind of organ or tissue in my body. So I cannot take from, I don't have a twin, I cannot take my immediate pedigree also not suitable. Somehow haplotypes are causing some issues where I have done for testing. So I have to opt to another match in terms of the allograft. So I might look for other human species. It's allograft. In humans, organ grafts from one individual to another are allografts unless the donor are identical twins. Unless isograft, all other are called allografts. And in terms xenografts, let's say you bring another same, not the same species. For humans, you get the grafts from either pig or either a baboon. These are called xenografts. Xeno is saying foreign or from space. Tissue transferred between different species. For example, baboon heart to human. This is called a xenograft. So fundamentally, grafts can be autograft, isograft, allograft, and xenograft. Allografts are foreign, right? Allo and xenografts are foreign. These two are nothing but self molecules only. No immune response will come. No immune illicit damage can be caused to these grafts given the selfness. But allo and xenografts will undergo a process of foreignness detection. They undergo a specific immune response. How the immune response will have in terms of tissue transplantation, genetically different individuals, allografts, allo means different, immune cells will see the MHC proteins of recipient as non-cells. For example, given the graft immune cells here, the green, they may see the recipient blue here as a non-cell because I don't know this MHC molecule. I haven't been exposed in my body. So what are these? I'm seeing as a foreign. So this will think worried about this. I'm in a new territory, new environment. What should I do? Should I kill them or otherwise I will be killed by them? So it will think of, it will be kind of shaken out. So it will take some response to that. So that's why matching should be done. Transplant donors are carefully selected by HLA antigens and also histone. If HLA identical individuals are also there, for example, monozygotic twins, we have seen that there is a major histocompatibility antigens. There might be a difference in minor histocompatibility antigen that may also cause a trigger immune response in very rare cases. Though the twins are here, they may get an immune response triggering because the minor histocompatibility antigens are different. So anyway, don't need to much about this. Concentrate on this MHC major molecules, how they will have. Thus, there is an immunological basis of graft rejection coming on from the graft. Where is the graft coming from? Allo or isograft, there is a graft rejection will happen. The degrees of immune response to a graft varies with the type of the graft. Self-graft or non-self-graft. Allo or xenograft, based on that, there is a graft rejection and a particular immunological response. So coming on to fundamentally again looking at the graft, it's a allograft transplant coming from two different species, two different humans of the same species. For example, humans, a mix of two are chimera. But though you look it as a different in terms of the color, but you look into the transplant here, basically if you take the donor and recipient out of the box here, there is this color, there is this color, two different grafts, for example, recipient. They're nothing but their cells, their tissues, their organs, their immune cells. It's nothing but the immune system. Immune system 1 here, immune system 2 here, which comprise different cells and tissues and organs and immune cells, leaving the donor and recipient, leaving the differences. As such, they are a kind of just we're looking at a person in terms of split personality. He looks different in a given scenario, but he looks odd in a given scenario. So the same split personality, you can just correlate in terms of the allograft transplantation, two kind of tissues having the same kind of fundamental immune cells or immune responses. They are not alike, they are different coming onto donor and recipient. The minute differences in their cells and immune system that will be exponentially increased by the immune system to protect one each other. Basically, you are looking at that's why you say, right, one heart transplant, you're looking for only it's a connective tissue. You can put only connective tissue. Then you compare the connective tissue of the donor and recipient, leaving all the immune cells taken off. It's nothing but just a blueprint of same, Xerox of the same. Connective tissue of the heart is same, given the donor or recipient. But it changes in terms of the what are the cells it's constitute of, where are the immune cells coming from, where are the blood coming from. It changes the different transplant, leaving that the transplants is nothing but a similar tissue. So basically given you're looking for a graft, you're looking at the recipient, you're looking at a different, it's nothing but these two charts are a kind of summary of the immune system as a whole. But the immune system in the graft coming from the living cell is nothing different from the immune system from the host. All same cells are here, giving the population coming in terms of the from the host. If certain subsidiaries populations, for example, T cells may be there, certain lymphoid organs may be there in the recipient, in the donor, and the recipient will have as such normal or normal functioning immune system. Basically, you're looking for one immune system is fighting over the other immune system. How do they fight? They fight nothing but they have their own mechanism. They have their own mechanism here. It's a tug of war between the foreignness. This recognizes the more foreign as this, it tries to elicit its own immune response. And this recognizes that as more foreign, dangerous to the self here. It will start its own immune. It will elicit it will trigger immune cells to go and attack it. It will trigger immune cells go and attack it. So there is a kind of tug of war scenario which wins over that. Based on that, you can say rejection mechanism is classified. For example, given a graft, given a host, the host immune cells from here, they went into the graft and they attacked the graft. Then they are nothing but host immune cells killing the graft. Host is dominant over here. The transplant is eventually rejected. For example, this modern warfare machinery is dominant here. It kills the graft. Basically, my immune system is very tough. I have a very slight foreignness also I can distinguish. I'm killing you. That's it. The graft will not be there, transplanted. So transplant rejection, host killing the graft. And the same way, what if the immune cells of the recipient are immunocompromised? They are not working proper. They are very sick. The immune system of the host is sick. Basically, in that scenario, the graft cannot be killed as per se. What if bad immune cells? What happens to transplant? Rejected or accepted? In most cases, it will be accepted without any problem, without any war between them. So they will be accepted just like that. Or in alternately, by intervention, external intervention, we might send some chemical agents to kill the host immune cells or to suppress the host immune cells for a time point so that the graft can come up here and the graft can establish itself in that donor, in the recipient. So that is called immunosuppression. This process is called immunosuppression. This is very important mechanism because the host kidney is non-functional, it's dying. So he or she needs to replace the kidney immediately to survive. In such a case, the graft will bring the kidney tissues. You kill the host immune with chemicals. The kidney will be transplanted. Then establishment will have, vascularization will have. The immunosuppression is also fundamentally important. So this is in terms of talking in terms of the host only. What if a scenario that the graft itself is highly dominant, brings in terms of the Khaleesi brings the two animals here, and then basically tries to dragons here, tries to kill the host. So highly dominant, highly efficient to kill the host. In that case, there is a kind of dominancy from the graft. It's going to the blue. The host killing the host immune cells is called graft immune cells killing the host. It's nothing but graft versus host reaction. It is also called GVHD disease. So basically, fundamentally, a host is dominant, a graft is dominant. You can get host transplant rejection. Graft can cause a disease in the host. That is the fundamental to the class today. Hopefully, given the time, we will cover, or we might have to do one more Zoom link for 5-10 minutes. We'll see that. So again, asking one fundamental question: how the host can accept or reject the graft? How it can do that? Basically, there is a fundamental mechanism. In a given scenario of here, 3 labels are here, 1 row here, 1, 2, row 2, row 3 here. Look into that. It's the first one is called autograft acceptance. So basically, I'm accepting the graft, autograft. So what will happen? I'm adding a graft here into a tissue, epidermis. Blood vessels are here, day 3 to 7 here. Within the start 0 days, within the 3 to 7 days, revascularization will happen. Blood vessels should come and re-establish themselves in a given new tissue. It's an autograft, so they can re-establish normally. And day 7 to 10, healing process will happen. Given the graft, given the cut and incisions, healing will happen. And 12 to 14 days, Resolution, normal flow of immune cells, normal blood, normal platelets, everything looks normal. And the given scenario of 14 to 17 days, normal vascularization will come in terms of autograft acceptance. So row 2, let's say an example of grafted epidermis, it may be an allograft here, and let's say a situation of it is rejected by the host, with the recipient. What will happen? Same, day 3 to 7, revascularization, same, it will happen. The blood cells will come and connect, but they will recognize it as a foreign non-self. Basically, the immune cells will get worried. The immune system will bring a lot of immune cells through clonal propagation, or signal will be multiplied. Multiple cells will come up here. The population of cells here, usually lymphocytes, phagocytes, and other inflammatory cells. They will basically tell that this graft is allograft. We don't need it, you kill it. So given a scenario of 12 to 14 days, the blood clots will form up here, necrotic tissue will be there, the graft will be rejected. So it's in a case of first case of rejection in terms of the allograft. But let's say the same graft is given to the same recipient again sometime later. So there is a kind of memory here. It doesn't need to go into the same response again. It quickly recognizes the tissue at the front. Within 3 to 4 days, cellular infiltration, blood clots, and all the necrosis will come in a shortened 5 to 6 days. It kills the graft. So basically self, non-self, how the establishment of graft and immune system, harmony between them is fundamentally important. So coming back in terms of HLA typing, what we are talking about, we look in terms of a system here, time after transplantation in terms of the months, a recipient has received a transplant, time after transplantation, 0 to 24 months, and how cumulative graft survival, whether the graft is rejected or accepted, you look into x-axis, y-axis here, you look into different combination of the arrows here, chart color, is nothing but you're looking for a kind of isograft or allograft. You're looking in HLA typing. You're looking for class 1, class 2 HLA typing. The curve 1 in green, see here, given that it's coming from class 1, class 2, almost HLA typing, no differences. Minor difference in terms of class 1. There is no issues in terms. The transplant will survive 24 months. Given a scenario orange here, you have class 1, 3 to 4 difference in HLA typing, class 2 only 1 to 2. You see the bar changes here given the 3 and 4 condition here, because MHC class 2 is really important. A change in haplotype from class 2 causes a difference in the variability. There is a kind of graph goes into this range, and this is here. And given a 5-6 condition, the survival of the graft is very less because a lot of mismatching of HLA types is happening in a combination of class 1, class 2. The graft cannot sustain here. So it's importance of HLA typing towards the graft that you can read in the text here. So how in general the graft will be established in the foreign territory, accepted or rejected, what is the fundamental mechanism of cellularization or vascularization or immune response. We look into HLA typing, how it's really important. So, okay, I will just pause it here now. We might have to again send one more link, bear with me.